Hi everyone, my name is Francesca Deleso-Frechette, and for my Laidlaw project, I studied the Cha1 treatment for triple-negative breast cancer. I chose to study breast cancer because my mom was diagnosed with triple-negative breast cancer when I was 5 years old, and she's now a 16-year survivor, but it is the most aggressive form of breast cancer, and so most people are not as fortunate as her. 1 in 8 women will be diagnosed with breast cancer at some time in their lives, and about 15 to 20% of those diagnoses are for triple-negative breast cancer or TNBC. So it is extremely prevalent in our society. Breast cancers are categorized based on 3 main characteristics: estrogen receptors, progesterone receptors, and HER2, which is another type of receptor. And triple-negative breast cancer is called triple-negative because it's missing all 3 of those. So if you picture the cancer cell as a locked house, in order to kill the cancer cell or get inside the house, you need to use one of these 3 characteristics or keys to get inside and kill the cancer cell. But triple-negative breast cancer does not have an increase in estrogen receptors, it does not have an increase in progesterone receptors, and it does not have an increase in HER2, so you can't use any of those treatment targets that are commonly used for other kinds, which leaves us only with radiation, chemotherapy, and surgery. My mentor, Amy Yee, together with her, all of the students working in her lab, came up with the Cha1 treatment, which is a combination treatment of epigallocatechin 3 gallate, or EGCG, which is a chemical found in green tea, and 5-azotiodeoxycytidine, or DIC, which is a type of chemotherapy. These are both given at very low doses, so they have minimal side effects, which will improve the patient's quality of life. We tested the treatment in mouse models, and as you can see, because the doses are low, each one on its own does not work. It has the same effect as the saline control group, but when they're used together, they complement each other and can decrease the tumor size. What my project was looking at was trying to figure out how this treatment works so that we can capitalize on that mechanism and bring the tumor size from that small size down to 0 by killing all of the cancer cells. The 3 main ways that I found that the Cha1 treatment works, together with help of the other students in the lab, was by increasing antigen presentation, increasing immune cell infiltration, and increasing treatment targets. So number 1, increasing antigen presentation. This is a normal cell, and the antigens on the outside are like tags, so the immune system can tell what type of cell it is. So this is a normal cell. We can call it a good cell. Say it's like a cell from your body. Obviously, your immune system does not want to kill it. But in a foreign cell, it will have different antigens, so then it will know it's like a bad cell that could be harmful, and it can kill it before it causes any harm. What happens in cancer is that a normal cell turns into a cancer cell. So it goes from a good cell to a bad cell, and it changes its antigens, but most importantly, it decreases them. It does this so that it can hide from the immune system and keep growing and growing, getting bigger and spreading throughout the body. The Cha1 treatment has increased the antigen presentation, so then it's more likely to be recognized by the immune system, so it's more likely to identify and kill those cells. The second way that the treatment works is by increasing immune infiltration. So in these pictures, the blue dots are nuclei, so each one is a separate cell, and the brown is around a blue dot shows an immune cell. So this is important that, as you can see, there's significantly increased, it looks kind of funny on the screen, but there's significantly increased immune cells in the tumor and the tumor environment with the treatment. And this is important because by proximity, if there's more immune cells closer to the tumor cells, they're more likely to recognize the tumor cells and kill them. And the third way that it works is by increasing treatment targets. So here, again, the blue is nuclei, each one is a separate cell, and the brown in this case is PD-L1. PD-L1 is kind of like antigens where it's a molecule on the outside of the cells, And this is really important that it increases it in the tumor cells because, like I was talking earlier about the 3 characteristics of cancer that can be targeted with other types of cancer, this creates a characteristic that can then be targeted with another type of treatment, such as an immunotherapy. Immunotherapy is definitely a promising future for cancer treatments, but so far the maximum response rate has been 19%, which is not very high. So cold tumors will not respond to immunotherapy treatments, and hot tumors will. And the Cha1 treatment changes the tumor from a cold tumor into a hot tumor by increasing antigen presentation, increasing immune cell infiltration, and increasing that treatment target, the PD-L1. So in the future, we could combine the Cha1 treatment together with an immunotherapy, such as immune checkpoint inhibitor, anti-PD-L1, and that would rewire the immune system to recognize the cancer cells and be able to kill them all, hopefully, curing the patient's cancer. This has an extremely promising future. We still need to test it together with the immunotherapy, but that's what I've been working on for my senior honors thesis. Thank you.
Are there any questions?
Yeah.
Really quick, do you know the underlying mechanism why the Cha1 treatment upregulates the...
Yes, it's very complicated. There's like a lot of different cell pathways, and we're not like, obviously we can't be entirely certain, but it increases interferon signaling, and we tested it by testing MHC class 1 and class 2, which are like on the outside of the cells. So that's what we think. Yeah. Yeah.
How do you select for upregulation
Such as we're still looking at that, but we're pretty sure that it only does target the tumor cells because we have 2 models. There's one that is a mouse that's immunocompromised that has a human triple-negative breast cancer tumor inside of it. So then we can tell easily which part is the tumor and which part is the environment because it's human or mouse. And the other model is the mouse version of triple-negative breast cancer, but it has a full immune system. So then it's harder to tell, but we need to look at the immune system so we can look at it there. But, yeah. Okay.
Thank you
Thanks.
very, very, very much.